In this video, we will discuss about how the proteins are trafficked towards lysosome. As we know, there are many diseases that are associated with the lysosome storage enzyme, and two of which that we have discussed in the previous videos. That is Tay-Sachs disease, and another one is Pompe disease. Both of which are related to the uh, dysfunctional of the enzyme that is present in lysosome, because of which the lysosomes and not been able to do its function normally. Let us understand how the first the proteins gets accumulated to the lysosome. This is the nucleus, and this is the endoplasmic reticulum. As we know that post-translation modification, specifically in glycosylation, begins in endoplasmic reticulum, and then it further extends or further modification done in Golgi apparatus or the Golgi complex. In the Golgi complex, along with the further modification and glycosylation, O glycosylation also occurs. So during this glycosylation or the post-translation modification, there are several carbohydrates or sugar or oligosaccharides attached to the proteins, and there is specific signals, specific kind of carbohydrates that is present in the protein that directs it towards the early endosome or the lysosome pathway, and that signature of the carbohydrates is mannose 6 phosphate. If mannose 6 phosphate is present on the protein, then it will be directed to the early lysosome or the lysosome pathway, early endosome or the lysosome pathway. And if the proteins that have mannose 6 phosphate escape this pathway, it will then escape this pathway that is shown here, will then move to the extracellular matrix, and in that case the mannose 6 phosphate receptor that are present on the cell surface membrane helps to capture those proteins and direct them towards the early endosome with the via endocytosis pathway. And after directing to the endosome, it will then move to the lysosome, where where the further breakdown of the protein occurs, and recycling of the amino acid and amino acid then recycled back to the cytosol of the cells. And while moving to the early endosome and lysosome, due to the acidification, the mannose 6 phosphate receptor and the mannose 6 phosphate receptor and the hydrolase and the protein hydrolase breakdown because of the acidification in the or the acid pH in the early endosome and lysosome, and the mannose 6 phosphate receptor will then move back or directs back to the cytosol of the cells, which will then retrieve more proteins with this mannose 6 phosphate receptor. So this is how the proteins move to the early endosomes and lysosomes. And this there are other processes also, such as vesicles that containing acid hydrolase will also move to the lysosome. And after lysosome, it will then goes to the proteins that are not able to uh, form their native structure, will then move. Some of them move via autophagosome, as we can see here. Microautophagy occurs just near the endoplasmic reticulum. It will then move to the autophagosome, and then it will then goes to the autolysosomes, and this lysosomes pathway will then collect both the lysosomes and autophagosomes, and then move to the extracellular matrix. And this is also the residual body, that is you can say that dead proteins or proteins that are non-usable by the cells are then moved to the extracellular matrix, where with the help of blood flow, it will then goes uh, secreted outside the body of the host. So let us understand in detail what are the proteins that are responsible for trafficking of the newly secreted proteins in the Golgi body towards lysosome. First one is the LIMP2. So LIMP2, then LIMP2 is one of the you can say one of the mannose 6 phosphate receptor that binds or the receptor that binds to the protein that has mannose 6 phosphate and then direct them towards the lysosome. And this similarly another proteins that is important is the PS1. PS1 also binds to the proteins that have mannose 6 phosphate and then directs them to the lysosome. And the lysosome also helps in maintaining the cholesterol homeostasis as it is shown here with the help of NPC1 and NPC2. And there are other things that are also mentioned also maintained by the lysosome is the iron homeostasis and pH homeostasis. As we know from a previous slide that acidification of the lysosome is important to recycle the mannose 6 phosphate receptor back to the cytosol of the cell so that they can take up more mannose 6 phosphate protein. And this and this V ATPase also helps along with the TRPML1 for pH homeostasis and autophagy and vesicle fusion, exocytosis maturation, and some of this that we have seen in the some of this we have seen in the previous slide. So I hope you will get some idea about how the trafficking to the lysosome occurs within the cells. And if you want to know more about this topic in detail, you can go to the reference that is provided in the description. So, thank you for your time. That's all for this video.